Hi, my name is Alyssa Villegas, and I'm in Dr. Eileen Anderson's lab, and I'm interested in the effect of C1q CD44 interactions on microglia phagocytosis in the context of spinal cord injuries. Spinal cord injuries affect approximately 15 million people worldwide. They're very debilitating, and they can happen to anyone. Now, after an injury, there is a lot of debris and cell death at the injury site. And microglia, the resident immune cell of the central nervous system, are the first responders to a spinal cord injury, and they are responsible for phagocytosing debris. Also present at the injury site are high levels of the protein called C1q, which tags debris for phagocytosis by the microglia. Now, Dr. Anderson's lab has identified 5 C1q receptors, one of them being CD44, which is a known macrophage phagocytic receptor. So, my question is, do C1q CD44 interactions regulate microglia phagocytosis? And to answer this, I cultured 2 lines of induced pluripotent stem cell derived microglia, one of them being wild type and the other being CD44 knockout, meaning it's missing the CD44 gene. To do this, I started with a low density of induced pluripotent stem cells. Then they undergo hematopoiesis, producing hematopoietic progenitor cells. Then those cells undergo microglial differentiation and maturation through the introduction of a cocktail of cytokines. Now, the whole process of going from induced pluripotent stem cells to a fully matured microglia cell takes approximately 40 days. So after I cultured my 2 lines, I plated for my experiment. I added C1q protein to the media, as well as pH photoparticles, and incubated for 1 hour in order to allow for phagocytosis to occur. pH photo are particles that fluoresce red in response to an acidic environment. So once the cell has ingested pH photoparticles, they will emit a red signal. Then I used flow cytometry and microscopy to visualize and analyze phagocytic activity. First, I used immunocytochemistry to validate my CD44 knockout line. And immunocytochemistry is a technique used to identify the presence of a specific antigen through the binding of antibodies. So here we have a wild-type microglia cell, and the green signal corresponds to the presence of my CD44 receptor. But when we look at the knockout cell, the green signal is missing, indicating that I have a true CD44 knockout cell line. Then I used microscopy to visualize microglial ingestion of pH photoparticles. As I mentioned earlier, pH photoparticles emit a red signal. So in A, the red signal is missing, meaning this cell did not eat pH photoparticles, while in B, the red signal is present, indicating that this cell ingested pH photoparticles. Lastly, I used flow cytometry to analyze phagocytic activity. So first, I gated for my cells of interest. Then I gated for single cells only. Then from that population, I looked at how many were positive for red fluorescence. And we can see that there are more CD44 knockout cells positive than there are wild-type cells. Next, I looked at the percentage of microglia phagocytosing, which is indicated by the y-axis, across different treatments of C1q, which is indicated by the x-axis. And indeed, we can see that the percentage of wild-type microglia phagocytosing increases as the concentration of C1q increases. In E, we can see the percentage of CD44 knockout microglia phagocytosing stays relatively the same throughout all C1q treatments. Now, when we compare the two together, we can see that the percentage of CD44 knockout microglia phagocytosing is increased compared to the wild-type microglia across all C1q treatments. In conclusion, CD44 knockout microglia exhibit elevated percentages of phagocytosis when compared to wild-type microglia. Wild-type microglia ingest pH photoparticles in a dose-dependent manner following C1q treatment, while CD44 knockout microglia do not show changes in phagocytosis rates. All of this leads me to believe that CD44 is a critical regulator of microglia phagocytic activity. My next step is to replicate this microglia phagocytosis experiment where I compare phagocytic activity for wild-type and CD44 knockout cell lines, but in a female cell line instead. Additionally, I would like to test a CD44 blockade as a potential therapeutic for not only spinal cord injuries, but as well as other neurodegenerative diseases. I'd like to take a second to acknowledge CERM, Dr. Monique Williams, and Dr. Brian Cummings for giving me the opportunity to do research through the CERM Compass program, as well as Dr. Eileen Anderson and Pooja Sakthaville for closely mentoring me through this project, and as well as the paper trail for giving me the opportunity to present my research. Here are my references, and thank you.